All right, new setup. Today, we are sitting down for an interview with ancient DNA expert Dr. Meradeth Snow. Please go ahead and introduce yourself and tell us a little bit about your background.
Awesome, thank you. I'm Dr. Meradeth Snow. I am a professor at the University of Montana, where I run the Snow Molecular Anthropology Lab, or the SMALL, with one L. It's very exciting. That was not my acronym at all, but it is stuck, so there we are. I have been here for 13 years. I'm a full professor, and God, what else about me? Let's see, got my PhD from UC Davis, and I originally am from California and moved here for work. And Montana is very beautiful, and that's what I'm going to say for that. Yeah, exactly. Wow, I'm being articulate today.
I wish I had some, drone footage of the mountains.
Yes.
Cut in there.
It's so beautiful. It's gorgeous. Yeah.
So, like we've gone over, your specialization is ancient DNA. So, what is ancient DNA?
Yeah, no, it's a great question because it's kind of this weird catch-all term, right? It's anything that's old, anything that's degraded, and where DNA crosses over from modern DNA to ancient DNA is highly contested. There's no firm answer on that. It really depends on the quality of the DNA, which sometimes you can have DNA that is not super old, but has been kept in conditions that were not very conducive to it surviving, so it functions the same way as ancient DNA. And therefore, it has all the same problems that ancient DNA does, which is basically where it's very short fragment sizes, it's seeing base damage, deamination, all sorts of other delightfully fun things that make it really difficult to work with. So you might think of it as just really horribly difficult DNA as the definition of what it actually is. It's just not a nice, beautiful cheek swab that you're going to be able to amplify everything and anything in. It's going to give you hell.
Right.
So,
yeah, I feel that.
I know full well that you understand exactly what I'm talking about.
Yep.
Yeah.
So, when we go and we do these studies on ancient DNA, what is possible with it, especially given that it is difficult to work with and it doesn't always work at all? So, what kinds of questions are we asking about, generally people from the past through their DNA?
Oh, yeah, definitely. I mean, there's lots of things you can ask. And as technology has gotten better, you can ask a lot of the same questions that we do of modern populations and peoples from past samples and past individuals and past populations. However, it's just a matter of whether or not there's enough DNA left, right? So, you can look at the genomes of human populations in the past and get great information that's very much correlates with what we have for today's populations. It was definitely going to have a smaller sample size, but it's going to allow for a lot of the same results if it works.
Right.
That's going to be the big issue, right? A lot of stuff just may not work. There may not be good DNA preservation, or it might be too degraded, or it might just be completely contaminated, all these sorts of things. So there's a lot of stuff that you can do, and you can look at past pathogens, which I know you know all about, and also then human genomes from the past, and also microbial genomes as well. If you wanted to look at the past gut microbiome or the past oral microbiome. So there's a lot of different stuff, a lot of really cool things that have been done recently, with even plants and other species, so not just humans. I know our lab focuses on a lot of human stuff or related stuff, but there's a whole world of other environmental or eDNA, ancient DNA, where you get it out of sediments, out of old plants, out of old whatever it might be, to allow for reconstructing past environments. Or my personal favorite, that talk that was at the essays this last year by Dr. Eski Willerslev, who was talking about utilizing past plant genomes to look at their adaptations to climate change, and then utilizing those same SNPs that they think were helping those plants survive in the past to put into plants today to help hopefully prepare us for climate change to come. So, which is kind of mind-blowing. So,
damn, I regret that I missed that talk.
Yeah, no, I think it was right before yours. I don't remember exactly how that worked out. It was crazy.
Yeah, it was overlapping with the session that my talk was in.
Yeah, totally.
So I was sitting in the back of the room, freaking out before my talk.
I understood entirely. Yeah, I was kind of like running down the hallway and like, oh yeah, okay, gotta catch Danielle's, gotta catch this other one. And then, yeah, but I really wanted to see that, and I'm really glad I did because, whoa, it was mind-boggling.
Yeah.
Cool.
So, where, besides your own lab, obviously, where can you do a DNA research? Where are kind of the institutional hotspots for this kind of work?
Oh my gosh, yeah, absolutely. So there's a whole bunch of different places. I will say that ancient DNA requires some very specific laboratories. So if you're going to go into this work and you're looking for schools that have this sort of capability, you're going to be looking for a very specific ancient DNA lab. There's obviously ones at Harvard, and then you also have one in Kansas. The one at Davis, actually where I got my PhD, is not working any longer. But there's Santa Cruz. Let's see here, where else? Max Planck, of course. Copenhagen, Leipzig, I believe. And yeah, there's a whole bunch of fun ones around the country. Yukon in Connecticut. Few of them, right? There's not every campus is going to have one.
Right.
There's a very select number of individuals that do this kind of work that are that masochistic to actually want to do this.
Yep.
Yep. You know what I mean? Anyhow, but yeah, so I would say that they exist, but they're not a huge number of them, but they're out there. And there's just finding those very specifically set up labs that have the contamination controls in place to be able to do this. So those are just a few of the places that I'm thinking of off the top of my head.
Right. So, and I think we've kind of touched on this a little bit kind of in your background, but how does a person become an ancient DNA expert?
Yeah. Oh my God. Some days I'm like, am I an expert? I don't know. I mean, I do this, right? Like,
I have also seen people kind of make the point that, maybe expert isn't the best word to use. Maybe it's more a specialist or a specialization.
That's fair. Totally.
Because there's so much to keep in our brains all at once, even within this rather niche discipline.
Oh my God. So true. Yeah. Then it's also like once you get into that niche, it like just goes forever. Like,
yes.
Yeah, like I don't terribly care about corn ancient DNA, but God knows that there's like entire labs that focus on that. And just look at the domestication of corn and like, it's really cool. Very fascinating results. Dude, so not in my wheelhouse. So yeah, not entirely what I want to spend my career on, but hey, it's all good.
But like what would the
but
kind of the path and the skill set that we gain along the way?
Absolutely. Yeah, so skill sets that you are going to want. I kind of came in this a bit of a like the back way, and I know you did kind of as well,
yes.
Like starting off a little bit more archaeology based, a little bit more history based, having these questions that you really couldn't answer with just the archaeology tools that are out there, like just the skeletal tools, just those sorts of things. Other anthropological fields can't answer the questions that you're really interested in. So that is where I immediately started. You're going to want a little bit of biology at the very least. Really a lot of biology. Honestly, today, I think to get into this field, you really do need to be taking your fair share of biology and genetics, chemistry. And I'm not saying you have to take like massive amounts of it, majoring or anything like that. Right. It might be helpful, but it's not necessary in terms of being actually able to do the work.
Right. Those are all the list of classes I did not take as an undergrad.
It is all good. I did, and don't know all the time if they entirely helped me. It's just, and maybe to that base knowledge somewhere along the line. But I think honestly, to get into this field, really you just need a lot of patience, maybe a little masochism, as I said.
Yep.
But yeah, I think being willing to learn, being open-minded, and understanding that you're probably not going to be able to take a straight line from A to B to C, which I think a lot of other, especially like strict biology working with the bacteria is going to have. You're going to have to find yourself, taking plan Z, incorporating it into plan F, and then maybe finding some other path to your results. It's never like, oh, you get the thing, you do the thing, and you get the data. It's always like, oh, you have to take a circumnavigating route around 10 different things. And maybe I'm not accurately describing a lot of biological research, but I think a lot of people like come into ancient DNA thinking, oh, well, it's just the same. No, it's not. Absolutely not. You can't use the same tools. You can't use the same kits. You can't use the same lab. You have to do all this other stuff that goes along with it that's so much more complicated.
I'm just thinking screams and PhD.
Yeah.
That all of that really tracks just with the process of doing a PhD in this. It's I'm not thinking about bioinformatics.
No, not at all. Oh, yeah, I guess that would be the other thing. Like, dude, learn how to code if you can ahead of time would be great.
Yeah.
Did I? No. Am I still trying to figure it out? Absolutely.
Same.
It's fine.
So as part of your, the way in which you train people to do this, the physical, get in the lab, do the things, you run lab camp every summer. So, and it has been a couple of years, since I went through this process, but what does lab camp look like? What is it, kind of the overview, the crash course?
It is the crash course, right? And like how to do the hands-on parts. Because I definitely noticed that like even my students that I start like graduate school even, that are coming from biology or some other field, and especially my students who come in from, strict just anthropology, don't know exactly like how to do a PCR, how to do electrophoresis, what goes into library prep for next generation sequencing. Like, you just don't get it. And you might have gotten it once in a lab and you had to memorize the steps, and that's like all you ever really saw. So lab camp is meant to provide the tools to go from, here's your protocol you might find either like on a website for a kit that you've bought or from like an article, to basically get you up to speed to implement that to some extent.
Right.
Is it going to be perfect? Probably not. It's going to take some work. But I start students at the very basics, how to pipette, how to measure stuff, right? Like the basic lab skills, and how to get DNA out of things, what that process actually is doing, like at a chemical level, so that like kind of can conceptualize it as opposed to just like following the steps in your SOP that kind of like, hey, this is what you do next. This is what you do next. Like, let's actually talk about that and like what it's doing, how it's working, and then translate that into actually utilizing that in not just a modern DNA lab, but then an ancient DNA lab with a clean room, protocols, and how to do all of that, and how then to get samples ready for NGS.
Right.
So yeah, and then at the very last day, we try to spend a little bit of time of like freaking everybody out on how to do bioinformatics.
I don't have a good segue into my next question.
So good.
Especially for me, going into hopefully my last year of my PhD and looking ahead to how do I find a job in this field, there's the question of what terminology do I use and what terminology do people like us, people who specialize in ancient DNA, use to describe our work. I've seen a bunch of different terms, and it might be different in the US, where we're coming through anthropology and biological anthropology and sometimes archaeology, a little bit different in Europe, but like all these different words and the different words that are out there, and like what terms do you prefer?
Right? Oh my God, right. Like what do I call myself? Oh my gosh. Well, I use molecular anthropologist primarily because it's now become incorporated into the name of my lab. And I feel like it hits on, like the genetic side of it, the molecular side, because I don't just do DNA molecules. Occasionally we get other stuff we have to do. Right. And then, we also have the anthropology. And I think for me, it's really important to emphasize both factors. So, or like an anthropological geneticist would also kind of work with incorporating both sides of what I do, because I think that they're both vastly important. Like, I could not do what I want to do and what I research without having an anthropological foundation. Like, I feel like that's so important. Like, what people are, how we act, what we do, where we come from. All of that foundational anthropological theory, really, that then plays into my genetics work. So I'm not just a geneticist. I am an anthropological one.
Genetics in a very specific context.
Exactly.
Yeah. Context being one of the single most important concepts you will ever need within archaeology.
So true. Yeah, absolutely.
Absolutely drilled into my mind as an undergrad.
Yeah, same. It's important.
Yeah. Yes. So, tell us a little bit about some of the current projects that you have going on. It seems like there's a lot, and I'm partly asking because I'm just nosy.
It's all good. Well, there's your project, right? Okay, so we have the projects in Poland, at Getch and Chisty Square. That's been super fun, looking at individuals, and particularly the pathogens, which have just, well, you've talked about that a lot, so I won't go too much in there. Yeah, but other places that I do work then is in Mexico. So primarily in northern Mexico, I like to work at the site of Paquime, which is in Chihuahua, Mexico, about 3 hours south from El Paso, which is a big city, pre-Columbian, super cool, lots of burials, a rich history, of course, of the population that lived there, but then also kind of like of the analyses that have been done in the last 50 some odd years, that have also allowed for us to have kind of like this massive network of information that we can build on and test and understand. And yeah, so particularly looking at like migration in that area and like population replacement and fluorescence of the city and things like that, which I've been doing that since grad school. It's super fun to me. I also work in West Mexico because we know that there were trade connections. If you were to look at Paquime and then the populations in West Mexico, we know that there was going to be trade of like shell and things like that, like lots of movement, at least, that we are assuming happened between these areas, but we're also trying to see if there was gene flow. Which has caused me untold headaches because a lot of the coastal sites are, speaking of terrible DNA, it's just so sad when you're like, oh, this tooth looks amazing, but there's like no DNA in it at all. Right. Of anything. It's not even like bacteria. It's just like, nope. But another thing, God, is a post-emancipation cemetery outside of Sugar Land, Texas. And that is where we have been utilizing the same skills that go into working with like ancient populations, but to work with the historic ones, because the DNA there again is highly degraded. It's not bountiful. It is going to have problems. It requires special techniques that are literally identical to what I do for like something that is there only like a little over a century old to something that's 9 centuries, right. 10 centuries old someplace else. We've been utilizing forensic investigative genetic genealogy to establish the identity of these individuals that were basically worked to death in this horrible situation of the post-emancipation work camps at the location. So that's been a field that I've been kind of delving more and more into, where I can utilize my same skills of working with just degraded and know how to work with degraded crappy DNA to then implement for forensic casework.
Right.
So, and then I could talk about all the forensic stuff too that we also do that kind of correlate with like sampling and the skeleton and how that plays into getting good DNA as opposed to like what often gets done in crime labs today, which is like just hack off a piece of bone and hope you have DNA in it.
Hopefully moving more towards into not needing to take as big a chunk of bone out of the remains.
Absolutely. If any at all, right? Like there's ways that we've been also working on for like the non-destructive methodology. We've got stuff to work, and then every once in a while you're like, why didn't that work?
Yeah. Again, complete lack of a segue into my next question, which I had thought about asking more about graduate school and the graduate school experience, particularly from the perspective of someone who oversees graduate students and that side of things. But honestly, that could probably be its entirely own interview.
Oh my God, right? Yeah, we should totally do a video on that.
I will never stop talking about grad school. So, what would be your advice for prospective graduate students?
Right. No, totally. I really liked your video about like email people because I know we were talking about it, and then I saw your little clip, and I was like, yep, absolutely. Please don't cold apply to my program.
Yeah.
I need to know who you are.
Yes.
I have been hardcore burned by situations where now I'm like, nope, I will have an interview at this point. Yeah. And I'm not gonna have time to do that without students who are interested in the program, interested in what I do. Hopefully you've looked into that. This is an assumption I'm making, which is probably not a good one. Then email me. I really swear I am actually really easy to talk to.
Yes.
Or try to be. I don't bite, maybe is a better way to put that. But I really do appreciate people who have looked into the lab, recognize our projects, or find something that they have looked into to me, the program, and are interested in and want to reach out. And so we can have a conversation. Tell me a little bit about why you're interested in the lab, what drew you to this, and don't just say that you want to be Bones from the TV show, Candace Brennan.
I don't actually know what her first name is. I think her surname is Brennan.
Yeah. But I should know that. But anyhow.
I have only ever watched one episode of the show.
Oh, yeah. Yeah, no, totally. I've watched more than that, which I'm slightly ashamed to admit, except it does make a very good drinking game. So, yeah. But yeah, no. Reach out, talk to me, look into the lab, do your homework. I don't want to just get an application in my pile of 60 some odd ones that I'm reading through for the next year. And I'm like, yeah, I don't know who this person is. You have to be a goddamn rock star for me to reach out to you. And that honestly has happened like once.
Wow.
So, otherwise I'm not accepting students that I haven't talked to before. I just honestly filter them out at this point. That sounds terrible, but as I said, I get like dozens and dozens of them, and I can take very few, and I'm really only interested in individuals who like know that I do ancient DNA. Have like maybe looked into what I'm doing, have reached out, have had a conversation with you, and that should be a Zoom or a phone call.
Right.
I recognize hopefully that from that you are not crazy. And that it's possible for us to have a project that we could work on together. And that does not have to be my work. I am fully open to like people bringing in projects, doing other things. Most of my graduate students end up coming in and actually putting together something that they actually want to work on. Very rarely do I be like, hey, I've got this thing here. You do this for your doctorate.
Right.
Like for your stuff. Like I knew you wanted to work in Europe. It took a little while to get things sorted. It's a bit of the post-pandemic insanity, but like getting that sorted out and getting you situated at the Polish sites was something that I knew you wanted to do, and we just made happen. So, yeah.
Right. And that actually, I should have put that on my list, a question about like, how do we even get these projects?
Yeah. That's a great question. Yeah, I think that it depends. Like Pakime or Poland, I'm interested in something, or I have a student who's really interested in something, and I will help with the legwork of like showing people how to approach people, cold call people, and be like, "Hey, I want to do this." I will though also note that a lot of my projects have just like literally fallen in my lap. Sugarland, which is this amazing, huge project that has like taken over my life recently in the best possible way. I love the people I work with. The community's amazing, so much fun, but literally came from a cold call on an afternoon where somebody was like trying to get some stuff situated with the DNA project inside of things, and some other stuff had fallen apart, and like they were like, we need somebody to do this. And if I hadn't answered my phone, or I don't think I would be involved in it. Yeah, somebody mentioned something as like, hey, let's do this. And like, oh yeah, hell yeah, that's exactly up my alley. Right. There gets to be a point in your career that also it's like, oh, you have funding for that? Let's talk. And then, yeah. So, totally.
Yeah. So what have been some career highlights for you?
Nature publications. I'm going to be honest. It was not poop, but it was still very cool. I still read that high some days. Other things in terms of research, which I kind of like different areas of my academic career, right? Like, have been returning names to individuals, either from cold case work or from Sugarland, or from not names necessarily, but being able to help with the repatriation process of Native American remains.
Right.
Like, it spoke to my heart in a way that like I don't think a lot of other things can. And I get a real like hopeful feeling when that sort of stuff comes together. It's not like pride. I want to do this work because I feel like it's very important, but it just is so heartwarming when it actually comes together and works, because it doesn't always. Like, you can't guarantee that you're going to get DNA out of something. But then there's the other things of highlights have definitely been seeing my grad students do fucking amazing things. Yeah, but no, I would say that being able to guide people, like, from like baby scientists, like scientists. I'm like, fuck yeah. That it honestly is probably the most rewarding part of my job. No, I don't think that. I know that. It's super, super important to me, and super a part of my, like, daily thought process of, like, okay, who needs what today? And, like, I do get spread way too thin, and I know I need to be better about that. But it is very, very much something that I value, and I wish I had a million more hours in the day to be able to do that more. It's just what I get my jollies from.
Kind of in the theme of, we're on YouTube doing some science communication, and I have personally done a lot of work, and I'm trying to continue doing work on the whole idea of using social media in particular as a vehicle for talking about things like ancient DNA and archaeology and anthropology. So are there any places where viewers can go find you on doing the social media science communication?
Yes, definitely. I do post quite a bit on TikTok, actually. I got involved with that a couple of years ago, had a couple of videos go viral, which was crazy. Never thought that would happen. I was like, what is going on with my life? Anyhow, hasn't happened since, but we're cool. Anyhow, but no, I really enjoy it. It allows me to talk a lot about what I do on the day-to-day, or things that I think especially get messed up with authors a lot, because I also, my other life is I write books. And one of the things that I just find myself absolutely annoyed beyond all belief is, like, when books, especially TV shows less so, I feel like TV and movies have some sort of, like, more leeway, I guess, in terms of, like, not portraying things super accurately because they've got, like, a time constraint. And I'm like, but books do not have that problem. And the research should have been done to portray it accurately. And it's so bad sometimes.
Yeah. I have a very specific example from a book I read years ago that comes to mind.
Oh, which one is it?
I do not remember the title of the book. And this was a strictly skeletal thing, and I think it was, they had flipped around limb bones. So there was a magic in the book, and they're like, oh, if you want to use magic to heal your broken arm, it's fibula whatever. I'm like, I about threw that book across the room because the fibula is not a bone in your arm.
I know.
And for me, that was something that is so easy to just Google a labeled illustration of the human skeleton.
Absolutely. No, I totally hear that. That would have made me probably put the book down. But yeah, no, mine, the one that honestly started me down, like, the path of being like, maybe if I'd make videos, like, for authors, right? Was the Discovery of Witches, that series, because Matthew in that series is supposed to be, like, a geneticist who's looking at, like, their creatures' DNA in the past, and I'm like,
that's not how that works.
That's not how that works. And then they started trying to talk about, like, the mitochondrial markers and all this other stuff, and I was like, not how that works at all. And then they tried to talk about sequencing, and I about lost my shit. And so, anyhow, it was so poorly done by a person who I, like, the author I view very favorably as a very well accomplished historian. I was like, somebody should have read your science bits.
Yeah. I have not read that book or
series.
Yeah,
no, I think there's like 5 or 6 of them now. I haven't read the last one. I do enjoy them. The TV show's terrible. But, yeah, that's like my alley of books that I really enjoy until I get to a part where I'm like, oh,
yeah,
there's so many other of us out there, right? That, like, I would happily read a short excerpt as an expert.
Yeah.
To kind of like go over and be like, hey, are you getting this like at least accurate enough to be passable to someone who is kind of the pedantic asshole that I am, that would not then be like, oh, for fuck's sake, this is not where you thought this would go, huh?
I was just thinking, I will put in text. We have plenty of space over our heads. I will put in, is it called a handle on TikTok? I don't know. I haven't been on TikTok in a while.
Oh gosh, I don't know. My handle works. I don't know if that is what it's called. I don't even think I ever knew what it's called. Your name on TikTok, your call sign.
I like call sign.
Yeah, I think that is actually the best way to describe it. But yeah, I am on there. I am also on Instagram, where I usually cross post my reels from TikTok.
Right.
And I have been on TikTok actually talking more about my books recently, because my publisher was like, why are you not talking about your books enough? And I was like, but I see, science to talk about is a little bit easier.
Yeah.
As opposed to being like, this is how I wrote this. And I'm always like, oh, yeah. Anyhow.
Gotta do that marketing.
I know. I hate marketing. It's fine.
Science is easier than marketing.
Mm-hmm. It is. Absolutely. Like 1,000 trillion%.
All right. Well, that is all of the questions that I had. And a lot of these questions came from not me. I did ask some people who are not themselves experts in ancient DNA, members of the general public, to submit questions for this. So there's a possibility maybe we could do that again in the future. But yeah, that is all the questions I had. Thank you so much for joining us here on Considering Skeletons.
Oh, yeah. Thank you so much for having me. It's so much fun. I've been loving watching how this is all like blooming.
Yes.
So, yeah, it's been great.
Mm-hmm.